Ah, não, acho que tinha uma janela separada, esqueci que não era uma aba. Meu Deus! Como é que era o nome mesmo?
É isso aqui, né? IBD, Covid, né?
Não.
É.
Isso. Pior. Não. É isso mesmo.
E aí ele tem um modo de visualizar.
Pode jogar no Chico, cara.
Mostra o Chico.
Beleza.
Não. Ainda não, porque aí agora tem que compartilhar. E o Márcio já se adiantou e fez o compartilhamento, que a gente está enrolado aqui. Desculpa, pessoal.
Ali apareceu. Vocês estão vendo aí direitinho?
Bem. Eu tô aqui. Bem, pessoal. Good afternoon, everybody. I would like to ask everybody now to be quiet because we are broadcasting it on our YouTube channel. And it's our great pleasure today to receive Dr. Marcelo Bozza for the opening presentation of these year's sessions of the Immuno webinars. As you guys know, Immuno webinars is a joint effort between the Biomedical Science Institute of the University of São Paulo and the Biophysics Institute at the University of Rio de Janeiro. And is co-organized by Gustavo Amarante Mendes at USP and me, Bruno Lourenço Dias, here at the Federal University of Rio de Janeiro. And count with many others help in this organization. And I should mention Cristiane and Miriam on our side, and what's the last name? Luisa. What's the last name again? I'm sorry, Luisa.
Menezes.
I'm sorry, Luisa. Luisa Menezes at USP. And I'll give you a brief introduction of Marcelo Bozza. Marcelo holds a degree in medicine from the State University of Rio de Janeiro, master's degree in parasite biology, and a PhD in cellular and molecular biology, both from the Oswaldo Cruz Foundation. He was a research fellow at the Harvard School of Public Health, and is currently a professor, full professor, at the Federal University of Rio de Janeiro at the Microbiology Institute, Paulo de Góes. Here at the university, he actively participates in the training of both undergraduate and graduate students, and he helped create and was the first coordinator of the postgraduate program in immunology and inflammation. That was the first immunology program in the state of Rio. And he was the head of the multidisciplinary network of computational biology for the study of host-parasite interaction with the support of CAPES, member of the immunology committee at the Brazilian National Research Council, the CNPq, and Secretary General of the Brazilian Society of Immunology. He received a chair, a CAPES Sorbonne chair, to teach and research at the National Museum of Natural History in Paris, and was also a Fulbright scholar at the New York University School of Medicine. He's a permanent member as well of the Brazilian Academy of Science. Marcelo has made seminal contributions in immunology with special emphasis on the studies of the role of heme and MIF in inflammation, and also on the studies of the pathogens of viral and protozoal infections. So it's really our great honor, both because Marcelo is a great researcher, a great speaker, and also a very good friend to us all. And so, Marcelo, the floor is yours. Thank you very much for accepting the invitation. And you have now Marcelo.
Okay, so thank you very much, Bruno, for your generous introduction. And it's a pleasure to be here. So many good friends in Biophysica and so many good friends at USP. So it's great. And I will present you today like two stories that are somehow, I think, connected. And the main axis of this connection is basically how we deal with infection. And in one way, through resistance, and through in a second way, a complementary way, through a disease tolerance. So I will try to show you using totally different scenarios how these issues are so important to deal with infection. No, acho que tá. That's all right.
Okay.
I think that I can move with the two computers here. So the general idea is that we really, as the animals, need to deal with infection in a daily basis for millions of years. And for each host, one can envisage these different possibilities that a specific pathogen cannot infect this host, or it can be a commensal, or it can be a pathobiont, or it can cause disease, and we call in this scenario a pathogen. And we have bacteria, fungi, virus, protozoa, parasites, and helminths as the main infectious organisms. And as I just mentioned to you, the way we deal with that is basically through these three different modes. I mean, here I present two of them, resistance and disease tolerance. And the idea of resistance is that basically it will impact the infection itself through controlling the pathogen burden. And this is mainly performed by the immune system. And all animals have immune system, innate immune system, and vertebrates will have, besides the innate immune system, also an adaptive immune system. And the ideal scenario of resistance would be that once you are infected, you have a reduction in your health. And if resistance is accomplished, you would be able to bring back to the initial state. So that's, of course, an ideal scenario. And we know that it doesn't work like this. So almost every infection, especially when we consider pathogens, they will eventually cause a damage. And this damage will be a direct effect of the pathogen or an effect from the immune system and the inflammatory response that might contribute to the tissue damage. So then we have a different way to deal with that. And this is the story of Mary Mellon that all of you might know. And the point is that she was a cooker in New York, and she transmitted a Salmonella typhimurium. Once eventually she was detected as the person that was transmitting the bacteria, and it told her that, look, you cannot cook anymore. And she stopped cooking for a while. She changed name and so on, and eventually start cooking again under another and a different name. And again, many people that were around her, eating her food, become sick again. And eventually she was isolated for life. And the main point here that I want to make is that actually she was healthy. I mean, she had the pathogen. To many people, that was a very dangerous pathogen, but she was completely healthy. She had, you know, good weight. She was not showing any signs or symptoms. So the idea then is that she would be someone that is tolerant to the presence of the pathogen. And this was originally demonstrated in plants, but also in different animals. And in this case, I show you some results from Janelle Iris and Ben Schneider on Drosophila. And basically what we see here using a wild type and a parental, a Drosophila. As you can see, the infection with a Salmonella would be more lethal, or at least a faster lethality in the wild type compared to the mutant. But the interesting point is that the amount of a bacteria was exactly the same. So in this case, a genetic difference is changing the way you deal with infection, but not changing the way you resist infection, not the way you control the amount or the number of pathogens. In this second scenario, again in Drosophila, what we see is that some of the Drosophilas would have Wolbachia as a commensal, and the model is the infection with this flockhouse virus. And as you can see here, the Drosophila that has commensals, the Wolbachia, they will survive, while the ones that do not have Wolbachia, they don't survive. And again, the important point here is that this difference is unrelated to the amount of bacteria, in this case virus, in these animals. So they have exactly the same resistant profile, but they have a totally different outcome of infection. So this is disease tolerance, and what is the general idea is that disease tolerance that is not immune tolerance, is not due to the adaptive immune system, what we call a tolerance, immunological tolerance, is mediated by T cells and so on. In this case, what we observe looking to these different individuals is that, again, health, pathogen burden, and in this case here, as you can see, in this arbitrary unity, this individual is healthy, he's going to work, he's doing, eating and so on. And this one shows now signs and symptoms of disease. And in this individual shows a severe disease signs and symptoms, and eventually at this amount of bacteria or virus, or these individuals have a totally different disease scenario. And in this case, this individual might be dying, and this one is still working and doing his thing. So as you can see, resistance is not the case here. It's not the main issue defining whether the person is or not healthy in these examples. Of course, that we know very well that the immune system is essential to keep health in infection conditions. And we know that because individuals without immune system or with profound immune deficiency will suffer from many diseases. So as I mentioned to you, the mechanisms related to disease tolerance are not so well known, but they are more related to the parenchyma cells and the different ways one cell or tissue will deal with the harm of the infection or the harm of the immune system fighting infection. And as you can see here, there are many systems that are related to these tissue damage control or cell damage control, and eventually allowing metabolic adaptation and survival of the cells and a health state, or eventually if these systems are not sufficient, you have more damage and eventually disease. So that's basically the way we deal with infection, as I mentioned to you, through resistance and disease tolerance. So one could say that in each individual host of a given species, the way we coordinate these two arms of a host protection will define the commensal community, the symbiotic microorganisms, as well as the infectious agents that are or tolerated or controlled, eliminated, or eventually able to cause disease. And so that is quite important, and it happens through a long evolutionary process. In the case of humans, we have another point that we have a short period of evolution that is related to cultural change. So if we look like 12, 13,000 years ago, we would live in a way that is quite different than we live nowadays. As you can see here, we would live in small groups, moving around and fighting for obtaining food. And we had what we call like the old friends. So we were mainly infected by these microorganisms. And this changed a lot after the agricultural revolution, when, as you can imagine, we start living in areas with more and more people, and also with animals, and in this case, transmission of infection, especially the ones that are transmitted by feces, for example, would start to be more and more important. And that was a long period, as you can see here. And eventually, there is a second major transition because of the Industrial Revolution. And the friends we have now might not be so friendly. And we, of course, would have a much more infection that are transmitted by orophycal transmission, but also we will have many infections with different viruses. And then we move to more contemporary time, and especially the Second World War II, it's a major change because we start living in a totally different way with very clean water and with an idea that is important to be hygienic. And we start also using antibiotics and vaccination and so on. So this again changed the microorganisms that we have, or the amount of these microorganisms interacting with us. So again, we turn to less helminths, less mycobacteria, hepatitis, and also more and more disturbed microbiota. As you well know, we know that the highly processed food will have a major impact in our microbiome. And it made a change. If we see, like from the Second World War II, we have a major reduction in several infectious organisms in the population, and we start having more and more of inflammatory disease, autoimmune disease, allergic disease, and so on, and also metabolic disease. And there is a huge correlation of these two things, and people believe that the hygiene would have a major impact on that. So we, among these diseases, the inflammatory bowel disease are very important, as you can see here, and it has a different etiological reasons, but we know very well that it depends from the genetic background together with environmental issues. So the point that I want to make here is that the inflammatory bowel disease is very dependent on the microorganism that lives in our guts. And of course, it's very much related as well to genetic susceptibilities. So we have several signs and symptoms, and we have long-term complications. And Prof. Bozza is a clinician and a collaborator in this study that has been studying IBD for many, many years. And as I mentioned to you, the paradigm of IBD pathogenesis is related to environmental factors, the intestinal microbiota, and the inflammatory response that it happens in susceptible individuals. And I won't go in details here, but as you know, the gut is a very complicated front for the immune system. As you can imagine, we deal with a huge amount of microorganisms all the time, and we deal with a huge amount of protein antigens that we eat. So we must have in the gut immune system a balance that would allow ingestion of these huge amount of proteins, living together with these bacteria that are the microbiome and the pathobionts and eventually pathogens, eventually pathogens as well. And we have an innate and adaptive immune system playing a major role controlling this whole situation. But in certain cases, we have changes in the environment of the gut because of antibiotics, diet, hygiene, pollutants, and infections. And these eventually cause dysbiosis and will change the community of these bacteria. And one important thing here is that the disease eventually can be transmitted from a sick host to a healthy host. And this is performed by the pathobionts and the pathogens present in the gut of someone with IBD. And as I mentioned, there are also several genes that are associated with a disease susceptibility. And one of them is IL-10 receptor and the IL-10 ligand as well, but the IL-10 receptor in children, it causes a mutation in the IL-10 receptor causes a very early onset of IBD in children. And it was shown many years ago in 93 that indeed IL-10 deficient mice will display IBD spontaneously. So if they are raised in a conventional system, they will show severe disease. It really depends. Oops. Yeah. Oh, yeah. I need to. Yeah. Cool. Thank you, Bruno. So the disease will be related to the presence of the microorganisms. So if the animal facility is a conventional one, the disease will have a more early onset and the disease is more severe. If it is a SPF, specific pathogen-free, the disease is less severe and it starts later. And if they are raised in a germ-free condition, there is no disease whatsoever. So the idea here is that these animals would have low tolerance to the presence of a somehow normal microbiome. And if we look in this figure, and this is not just to IBD, I mean, this is something that holds for many, many different infection conditions. The IL-10 deficient mice sometimes might even have a better resistance, as you can see here, with a lower pathogen burden. They have more disease than a wild type. So indicating that they really have low tolerance. We have been studying macrophage migration inhibitory factor for many years, since my PhD, when I developed the MIF deficient mice. And nowadays, we know that it's a pro-inflammatory cytokine that binds to different receptors, the invariant chain of MHC and CXCR4. It controls the expression of different TLRs, including TLR4, and it inhibits apoptosis, and it maintains macrophages and neutrophils in the inflammatory sites. So we have been studying, as I mentioned, for many years, and one thing that we notice is that for many infectious models, MIF would be pretty much the opposite of IL-10. So the MIF deficient mice would have a better health state compared to the wild type in many infectious models, even though in some of them with lower resistance. So it would be somehow the opposite of what we see with IL-10. And one example is this model of a oral toxoplasma infection. And as you can see here, and we did this study already many years ago, but what we observed was that MIF would have a better survival compared to the wild type, and the pathology was better in these animals. They had a less reduction in terms of the gut, and less immunopathological score, as you can see here, comparing these. But what is interesting, as you can observe, the MIF deficient animal had all these parameters preserved despite more a pathogen in the gut. Another study using a T-cell transfer model of IBD also demonstrate that MIF deficient mice has a better disease response, as you can see here. And this is not due to the MIF present in T cells, but in other cells than T cells. And also treatment with anti-MIF, similar to the treatment with anti-TNF, prevent, and also has an impact, a therapeutic impact. So, and in this model of cell transfer, and also in this model, there is a chemical-induced disease. So, what we did was basically to ask the following questions. I mean, they are more or less the same question made in different ways. So one is high or low tolerance to gut inflammation dominant in IBD, or is MIF involved on IBD development of IL-10 deficient mice? So that was basically the questions we had. Was a very naive idea. And what we did basically was our plan was to obtain a double knockout. So we would see which one would be more important, would be dominant, whether the lack of MIF, the lack of IL-10. And so basically, I asked my student at that time, that she just arrived in the lab, so I said, well, let's start crossing these animals. And after 10 days, she came to me, Poliana, she came to me and said, oh, something really weird is happening. All the IL-10 mice that were in the cage with the MIF mice died. And I said, okay, that's interesting. So why don't you go again and now do it in a scientific way? So you go there every day, you weigh them, you see when they die, and so on. So that's what she did, and she started doing that, and that's basically the design. As you can see, a very simple experimental design. So we would have IL-10 mice alone, IL-10 mice with 1 wild type and IL-10 mice with 1 MIF mice. And we have like several boxes like these for each combination. It's something happened? No. So and then what we observed was that indeed so here, just to let you know, all animals are IL-10 deficient mice, okay? The IL-10 deficient mice together with IL-10 deficient mice that was kept together with wild type, and IL-10 deficient mice that were kept with MIF deficient mice. But all animals we see here are IL-10 deficient mice. And as you can see, they lose weight and they eventually die. The ones that were together with the wild type or with the IL-10, they were fine. The ones that show disease, as you can see, the rectal prolapsum and eventually blood in the feces, and of course they lose a lot of weight and so on. So we look better to what was going on. We didn't know exactly what was going on. Of course, the bleed in the feces, the rectal prolapsum all suggest that they were going through an inflammatory bowel disease. And when we perform an analysis of these columns by video, as you can see, again, all of them are IL-10 deficient mice, and the ones that were just kept with IL-10, the ones that were maintained with wild type, and the ones that were maintained with MIF deficient mice. As you can see, they have like these inflammatory and bloody aspects. They also had like reduction in the size of the column, and they have changes in the histology. So we analyzed the populations in the lamina propria, and we observed that the IL-10 deficient mice, again, it's always like this, okay, at this point. So we are looking just IL-10 deficient mice. So as you can see, the ones that were IL-10 with IL-10, the ones that are IL-10 with MIF, and the ones that are IL-10 with wild type, they have an important increase on CD4 gamma interferon positive cells, and we don't see a significant increase in these other parameters, although it seems that the MIF, the ones that were kept with MIF, they start showing more of the double knockout, but it's not a significant. The double interferon and IL-17. And we look the neutrophils, and also we observe an important increase in this population in the IL-10 deficient mice that were maintained with MIF. And we analyzed the FOXP3 population. It is also increased in the IL-10 deficient mice that were maintained with MIF, but of course these regulatory T cells are unable to produce IL-10, and they were not sufficient to hold the inflammatory response. So I don't know. I mean, if you want to ask questions, I think that it's allowed, right? I mean, yes. You guys can interrupt me and ask questions. We haven't looked, but it would be cool to take a look, Julio.
So I just ask you to repeat the question so people in São Paulo can also hear the question. That you
if you guys, okay.
Because from here, you probably cannot hear.
Okay. Yeah. Julio asked whether we analyze a gamma delta T cells. I think that's a very good point. That we would love to do it, but we haven't done it yet. So then we had another question. you know, at this point of the project, whether this disease transfer was not really a disease transfer, but a disease that was triggered by the presence of MIF was related to the presence of an infectious agent. So that was, but it could be a virus, it could be a bacteria, it could be many things. But our hypothesis at this point was that animals of a tolerant genotype to intestinal damage, that is the case of MIF, might harbor a microbiome that is enriched in pathogens or pathobionts. So that was our new hypothesis. So we then perform again a simple experiment. We collect feces from the different genotypes and put in the cage of the MIF, of the IL-10 deficient mice. So basically, that's it, like feces from wild type, MIF. And again, as you can see, the animals that receive the feces of MIF deficient mice lose weight and die. And it's important to tell you that mice is coprophagic, so they actually eat feces. So, and they become sick. And when we actually filtrate the feces from the MIF deficient mice, we observe that this feces now is unable to provoke disease. So in this case, the filter would block bacteria, but not, and fungi, but not virus, or metabolites. That would also be a possibility. So we analyze the microbiome of these different genotypes. And the idea here would be, as I mentioned to you, that, well, by being more tolerant, maybe they have a more diverse microbiome. And that's exactly what we found. As you can see here, the IL-10 deficient animals have a very homogeneous microbiome, that cluster here, and the MIF and the wild type, they have some clusters, but they are more or less in the same area in this PCA. But when we look the alpha diversity, as you can see here, the IL-10 has less diversity, the wild type and MIF has more diversity, exactly as we thought. And we look the Firmicutes and Bacteroidetes that are considered related to IBD. And what is interesting at this point of life of these animals, and just to make it clear, I mean, here we are analyzing the microbiome before any co-housing. So we are just looking the microbiome of the different genotypes before co-housing. And what is kind of interesting is that the IL-10 deficient mice, although we know that they would become sick just with time in the cages, they have a better bacterial profile or a more healthy bacterial profile compared to MIF. So here we are collecting around 6 and 8 weeks. And so we ask why these guys have this different a bacterial profile, and in this case we look the antibody response in the gut. Actually, we look the antibodies that were covering the bacteria. And basically what we observed was that the IL-10 deficient mice, they do have more IgM and IgA coated bacteria compared to MIF and wild type. And as you can see here, they have more IgM, a single positive bacteria. And with more IgM per cell, per bacterial cell, and the same thing concerning IgA. So the IL-10 deficient mice seems to have a robust antibody response to the bacteria in the gut. So we want to look in the future experiments. It's not a very easy experiment. What would happen if we transfer IgM-coated, IgA-coated, or uncoated bacteria? So which one of these groups would be the more related to the disease transfer? We also look to the epithelium of these, the gut in the column, but also epithelium mediators, alarmins and defensins. And what we basically found different was that the MIF deficient mice compared to the wild type, they have more IL-33. There is a cytokine produced by epithelial cells. They also have more IL-22. There is a cytokine produced by especially innate lymphoid cells and are associated with epithelial protection. But we haven't seen major difference in other mediators. So we know at this point that the adaptive immune system is a bit different, comparing the genotypes, and comparing the wild type and MIF. It seems also that eventually the ability to regenerate the epithelium might be better in the MIF deficient animals, but we don't know exactly what is the importance of this. So as expected, after the co-housing, the diversity of the microbiome of the IL-10 deficient mice increased. So it seems that they received the microbiome of the MIF deficient animals. And we, of course, decided to treat with antibiotics. And as you can see here, this cocktail was not able to block the disease, the start of the disease and the disease in the IL-10 deficient mice that were kept with MIF. And we did these experiments in several different ways, like treating both MIF and IL-10, treating just MIF, treating just IL-10, treating during, before and during the co-housing, and we were unable to block disease initiation. We isolate some bacteria in the feces of IL-10 deficient mice after a treatment with antibiotic and after co-housing with MIF. We had the expectation that these different consortiums would eventually cause disease. We did one experiment was just before the pandemic, and we used IL-10 that was germ-free. So it's maybe it was supposed to be an ideal experiment, but it didn't work. So we don't know whether the fact that they were germ-free, it changed the ability of become sick in these a transfer of bacteria from that were isolated from the guts of IL-10 after co-housing and antibiotic treatment. But then we were able to obtain a double knockout, so it wasn't that easy, but we ended up using a knockout of MIF with heterozygous of IL-10, and we were able to generate some animals. But as you can see here, that would be the expected number in a Mendelian ratio. And as you can see, so we obtained in this crossing, as I mentioned to you, this is MIF with both alleles deficient, and IL-10 with one allele. So we cross them, and that would be the expected ratio. And basically what you can observe here is that the double knockout would come in much less number, and very early they start showing the rectal prolapsion and hemorrhage in the colon. So at this point, we don't know exactly what is going on, that they die that early. But as you can see here, if we co-house these double knockouts with the IL-10 deficient animal, they also are able to trigger disease in the IL-10 deficient animal. And the same thing, if we transfer feces from the double knockout, we also can trigger disease in the IL-10 deficient mice, indicating that the microbiome of this double knockout is somehow similar to the microbiome of MIF deficient mice. But they don't deal well with the presence of this microbiome. So the double knockout seems to have the microbiome of MIF and the low tolerance of the IL-10 deficient mice. Well, that's a neat. So the question of Miriam is whether these animals are kept SPF. So no, they are not kept SPF. They are kept here in our facility. But the good point is that the IL-10 deficient mice, it takes very long to become sick. So indicating that our facility, it looks like an SPF, at least considering the kinetics of the disease in IL-10 deficient animals. So, but we know they are not SPF, and I think that probably why it show up the phenotype.
Maybe you should try doing the other way around, the IL-10 knockout with MIF heterozygous, because maybe the MIF knockout keeps their...
Yeah, but that's the problem. Yeah, the problem is that they die.
Even
yeah, if they are like the knockouts, they will die, right? But that's an important point. So the mother would be ideal to, once we should do a C-section and have a foster mother to keep the pregnancy. That would be cool. So the conclusions of this first part, I don't know, what time is it?
1 o'clock.
1 o'clock. So I don't know, we go fast. I think the other is very short.
Yeah.
So the conclusions are basically what we just discussed. So there is no major different conclusions to the point we just discussed. So the co-housing triggers colliding the IL-10, wild type do not, and also the wild type do not become sick when co-housed with MIF, indicating that the genetic susceptibility of the IL-10, it's not there in the wild type, of course, and it's not and is essential to the triggering of the disease. So the wild type can stay with MIF and they are not sick. So as we saw, the feces promote a disease, but when they are filtered and the bacteria and fungi are not present, they are unable to promote disease. So our hypothesis that a more tolerant animal has increased diversity of the microbiome was shown. So we confirmed the hypothesis. And we also observed the presence of pathobionts that are able to promote disease. We don't know the name of these pathobionts. So the antibiotic treatment was unable to block disease, and we isolate some pathobionts that could be candidates to promote disease. So we believe that low tolerance is dominant to disease development. So the lack of IL-10 in the double knockout is still prevail in terms of the disease development, while the high tolerance, it seems to be dominant or defining the microbiota. So it's, as Miriam mentioned, of course, that in this case, we have mice that were in the cage when the double knockout were born. So it would be interesting really to do a C-section and transfer the animals to a foster mother. One thing that I didn't mention to you, and I think is really important, or I mentioned in the very beginning, is that there are many models that mice that have IBD can transfer disease through the microbiome. But as far as we know, this is the first model that a healthy animal with even a better way to deal with IBD transfer disease to a susceptible animal. So the hypothesis here is that individuals that are more tolerant to a tissue damage in the gut, they might harbor a community that has the ability to cause disease in less tolerant individuals. And it's possible that in families, like more tolerant relatives would promote or trigger disease in susceptible individuals, in child, through pathobionts. And we are like interested to understand what are the bugs associated with that, and why exactly MIF deficient mice would be more tolerant to a gut inflammatory damage. So I want to thank people in this first part of the talk. So Vinicius is the person that got this project in the very beginning when Poliana decided to do medical school. So she decided not to stay in the PhD, and Vini started these experiments in the very beginning. And Leticia was a master student, now a PhD student that was associated with Vinicius in these studies. Jamil helped setting and performing the initial experiments of bacterial coating. Now we did all again, and Elena helped a lot with the flow cytometry, and Andressa helped with the histopathology. Heitor was essential in the different analysis, the video, the video in the column, the histopathology. Dan Littman and Varsha did the isolation of the bacteria after antibiotic treatment and co-housing. Fabio, it's also very important in the flow cytometry analysis in the gut and in the whole project. He is together with me supervising Vinicius, and Batia, he was essential to all the microbiome sequencing and analysis. So now I move to a different story that we were involved because of the pandemic. So one of our questions here, it was eventually asked whether it would be possible to have very early markers of a resistance and disease tolerance in COVID. So basically, we know pretty much to almost all disease, with some exceptions, that when you have an infection, you might have people that will show absence of disease signs or symptoms. Some people will show mild disease, some will have a more severe, eventually needing oxygen, and some will have critical outcomes. And as you can see here, this was an early study, but basically, as you can see, like the more severe cases are among like 15–20% in terms of in COVID infection. So I don't need to show you that, but we all know what are the main players in terms of resistance to viral infection. And we have the innate immune response, the adaptive immune response. So we, and the communities have been studying these different parameters due to COVID. And as you can see in these two figures, in one of them, we see a response that are dominated by type 1 interferon, and eventually helps to hold the viral burden, and the adaptive immune system with CD4, CD8 cells and B cells producing neutralizing antibodies. And if all goes well, this individual will have a more mild response. And in the case of in the severe cases, we see that this response of type 1 interferon might be delayed and sustained later, and many inflammatory mediators would contribute to the tissue damage observed in the lungs. So it would have like a sort of like cytokine storm and so on that was well documented. So here I show you again the idea of a resistance and tolerance, giving like the four major possibilities. Imagine that someone has high resistance and high tolerance. That would be the best scenario, right? So you eliminate the virus and you don't have much damage. You might have high resistance but low tolerance. So in this case, you clear the pathogen, but that might have a cost. It really will depend. Let's say if you have a vaccinated person, the resistance is high and eventually high enough that you don't see a disease. But in a natural condition, one can imagine that having high resistance do not guarantee that you won't be sick. So if you are low tolerance, you might still be sick, although you clear the virus. Another possibility is that you have low resistance, but you have high tolerance. So in this case, most likely the patient would have a persistent infection and would be asymptomatic or had a mild disease. And of course, the worst scenario would be low resistance and low tolerance. So you would have a lot of virus and a lot of tissue damage. So that was more or less our way of seeing this a problem when we were able to start studying COVID. And the main aim was to establish a correlation between a resistance or the immunological parameters associated with resistance and disease tolerance profile of early infected individuals. And by early, I mean when they came for the first test of COVID. So, and this was performed here at the CTD, and that was the workflow. So we would receive, the patients would have the interview, blood collection, the swab, and the samples would come to prepare a plasma and PBMC, and the team would also, in this case, sera was used to do the antibody titers and the extraction of RNAs from the swab for diagnosis. And our team would do like the analysis from the PBMC, and also we collect plasma. So we had different groups, and that was done like this. So we collected the sample of the patients at day, let's say 3, at day 5 of symptoms. And we maintain contact with the patients. So the patients would say, look, or the family would say, oh, he went to hospital. Oh, he's okay. So, but all of them in principle would come back to see whether or not they were negative of the infection. So this was done here at UFRJ with many of the people from our community. So we had a close proximity with the patients. So we collect in the very early time point, and we knew what was going on with these patients, and eventually we had these different groups. So patients that were oligosymptomatic and were able to clear infection up to 21 days. Oops. Patients that were oligosymptomatic, but they were still positive after 21 days. Some of them would be positive for more than 100 days. And the swab of some of these patients were able to infect target cells in vitro. And we also had patients that had an unfavorable evolution, so they eventually require oxygen therapy. But again, these patients, they were walking just as all the other patients when we collect these samples. So they had no severe disease at this time point. So we again asked the question, is it possible to find something different in these different groups in the first sample collected? So that was more or less the proposition. So this is the CT, the cutoff of the CT. And as I mentioned to you, like the regular, persistent, and severe infection. And of course, this is not a hospital, so we didn't have a lot of severe cases. We have more than that we are analyzing now, but the numbers are not huge because we collect the samples when they were just like all the other patients. And we did the different analysis, and I will show just some of them to you today. Here, the CT values, and as you can see, the severe, it's not significant different, but for sure, they are not more resistant, or they are not more susceptible to infection, in the sense that they even seems to be more resistant than the other groups. So the severe case, one would expect that they might have much more virus. That's not the case. And this is the kinetics, as you can see. So we were able to collect samples in different time points until they become negative. So this is the regular one. So finishing by 21 days, the persistent ones, and we have certain individuals, as I mentioned, with more than 100 days of infection. And the severe, we had some with some persistence and some that clear infection, just like the regular ones. And just showing here the non-persistent and the persistent ones. As you can see, here is like when they still were positive, and the white one when they completely negativate of the virus. We then analyze the different parameters as we discuss some of the cytokines and mediators that are related to this initial phase of the innate response. So the interferons, we analyze also the plasmacytoid DCs. And as you can see, comparing these non-infected, the ones that we call regular, to the lack of a better name, the ones that are persistent, and the ones that are with severe infection. And as you can see here, the number of PDCs, it's less in the persistent and in the severe. The interferon, it's also reduced. It's not significant, but there is a tendency of reduction in this case. And the severe are producing a type 1 interferon, as you can see here. So the severe case, they are not lacking a interferon. And we also see a auto-anti-interferon alpha in these individuals. The severe ones have more autoantibodies again against a type 1 interferon. They have more IP10. This is a gene that is induced by the interferon, the type 1 interferon. And again, as you can see here, the regular one, the persistent, and the severe cases. And also when we look to the transcription factor, IRF7, they are again increased in these two groups, but not in the persistent patients. And these ISG also is not. So the point here, it seems like the persistent ones have less type 1 interferon response compared to the regular one and to the severe cases.
Marcelo, quick question. They are free of treatment because you would guess that the severe patient would have received whatever.
Yeah, no, because it's very early.
Right?
This is very early.
Even the persistent one is also free of treatment. Right?
Yeah, because this is all like the first collection time. So we didn't even know whether they were positive or not.
But you don't know also if they were treated at home, home treatment.
Well, we have these in the interviews, whether they took, let's say, chloroquine or but ivermectin. But again, remember that all these patients are outpatients, so they came here to do the test. So of course that some people would start treatment even before positivation, but that's not the common point, right? So the majority of individuals would first ask, am I infected or not? So that's why they came here to do the test. So that was before any treatment.
Marcelo, so this means that those individuals were grouped like in a post-hoc range.
Exactly.
Not characterized further.
That's exactly the thing. So we kept the samples. We kept doing the diagnose tests, and if they were positive, we collect blood and did the PBMC and so on. But that's exactly the point. That it was all analyzed afterwards when we knew the outcomes. And then I don't know if you guys can see, it might be a bit small here, but basically what we looked was the type 1, type 2, and type 3 here called based in Th17 type of response. And I think that result is kind of interesting. Again, I mean, it's very early time point, but as you can see here, the persistent and the severe patient have less a type 1 immunity. As you can see, they have a less gamma interferon and less IL-12 compared to the regular ones. The type 3, as you can see here as well, they are a increase in the severe patients and in the persistent patients compared to the regular one. So they seem to have less type 1, more Th17 type of response, and it's known that severe case has more neutrophils. But what was really nice to see was that, they both moved together, like less type 1, more type 3. But then when we look type 2 response, and Carolina Lucas had shown before that the severe cases that were from patients that had the samples collected a little bit later than ours, they had this shift So the severe patients in her study had less type 1, more type 2, and more Th17. And we confirmed that. So the severe patients have less type 1, and they have more of type 2. And here, if you see IL-9, I think it's an interesting marker because, as you can see here, in the case of the persistent patients, they do not have the increase in eotaxin, they do not have increase in IL-13, and especially IL-9, they have even less than the regular ones and much, much less than the severe ones. We don't know exactly what that means, so we would love to do a mouse experiment to see what's happening if you block or if you give IL-9 to the animals. So our expectation was that the persistent ones would have more IL-10 or more TGF or more of the tolerance-associated genes, such as NRF2 and homoxygenase 1. But that's not the case. As you can see here, oops, the persistent and the severe group patients have less than, and the ones that we thought would have less of NRF2 and homoxygenase, they actually have more. So indicating that they have a response that it is somehow working to hold that inflammation, but that is very early, as I mentioned to you, but it doesn't seem to be sufficient. So they are healthy at this moment, but they will become sick after 3-5 days after this time point. So yeah, we analyzed different parameters associated with tissue damage and tissue repair. And overall, the impression we had is that it's like the persistent were not even infected, or they were more like, in some cases, similar to the non-infected than to the regular ones. So they had less response of everything. And the markers like of tissue repair, they are increased in the persistent ones, in the severe ones. Here I show you some results of the persistent. We are finishing to do the same in the severe cases. Basically, we analyze whether they had a defect in terms of adaptive immunity. So we analyze a CD4, we analyze CD8, and also, as you will see, the antibody response. And basically, this is a re-stimulation with a SARS-CoV-2 peptides to show that they don't have any major difference compared to the now non-persistent ones or regular ones, but the reduction of IL-10 that we had shown systemically. And again, against our initial hypothesis that IL-10 could be increased. And they also have more IL-21 in the CD4, CXCR5, TFH compartment. And when we look the antibody response, as you can see here, comparing, in this case, non-persistent and persistent, we don't see a major difference here, both in IgG and IgA. But what is kind of funny is in this assay, there is a neutralizing a surrogate test by competition. What we see is that the persistent ones have especially in the starting here, but especially the convalescent phase, they actually do have more neutralizing antibody against SARS-CoV-2. Again, against our hypothesis that eventually they would not be able to have, a good antibody response. and they will not be able to clear infection. It doesn't seem to be due to antibody response. And in fact, many studies have shown that severe patients also have more of a neutralizing antibody against SARS-CoV-2. So we have still more questions than answers, but what really it made like an impression to me, it was that very early you can separate the severe, the regular ones, and the persistent ones based in some markers, especially the type 1 and the type 2 response. So just to summarize here, so the persistent ones would have, compared to the regular ones, less type 1 interferon. I didn't show all the results. They have less of some of the monocytes subpopulations, and they have less IL-9. I'm not sure whether this would be a TH9, but it could be TH9 the source. When we compare the persistent and the severe, so both have reduction on type 1, both have increase in type 3, and both have reduction in IL-10 production. And the severe ones, at least in that experiment, they seem to have similar or less viral titers. They have more of the stress response genes and tissue repair molecules, and they do have more IL-9 production. So I want to thank the people that did this study. So this persistent, the study with the persistence was submitted, is under review, and Elena is the person that is the first author in this study. And we also have Victoria that is doing the MD PhD, and she's in Akiko and Carolina's lab right now, studying long COVID. Guilherme is also a PhD student. Clarice was a postdoc. She's now at NYU. João is an undergrad student that I didn't show most of his data, but he did a lot of analysis. And this is in the manuscript, but not here. Heine is a master student that did the analysis of cytokines and the expression of NRF2 and type 1 interferon genes and so on. So all the group work a lot to isolate the PBMCs and to follow the patients and so on. And of course, this study was performed with a lot of people from different groups, and André Vale, Terezinha Catineiras, and Juliana Echevarria were key to this project. So this one group of results of a project that will still have many more things to come, and André for sure will eventually present some of his very beautiful experiments. And Dominique was a French collaborator that together with me were able to obtain this financial support. So I want to thank you all for your time and patience.
Well, thank you very much, Marcelo. It was a very exciting presentation. As is our tradition at Immuno webinars, we are going to open the questions first for the students. If any students here or watching online would like to ask some questions,
Go ahead.
No.
Come here. Oh, yeah.
We have a question here from Lucas Bolini, who is a PhD student here at the Federal University. Come close here so you can.
Okay.
So, very nice talk, Marcelo.
Thank you.
I was wondering, regarding the first part of the presentation about the MIF knockout mice, you showed that there's less coding of IgM and IgA on the bacteria. I was wondering if the MIF knockout itself would induce some hindrance on the production of those antibodies or on B cell recruitment or activation or anything like that. Like, how would the MIF knockout affect the coding itself, or if it promotes a class switch, because you didn't see anything about IgG or IgG subtypes coding bacteria?
But yeah, that's a really cool point. I mean, our initial idea is that the IL-10, because the barrier dysfunction would have more of a adaptive immune response. Conversely, the MIF, what we think is that they might have more of an epithelial, a healthy program. So the damage would be repaired faster and so on. They would have less bacteria entering, activating the adaptive immune system. But it is true that the MIF and wild type, they are not as some.
Pessoal, acho que congelou. Caiu.
So,
yeah, just a second. We had a small glitch in our internet connection for everybody here, but it's back already. I'm sorry for this.
So yeah, we are starting analyzing the B cells, and I think that we're going to have this answer shortly. Thank you, Lucas.
Okay. Is there a question on for people that are watching the streaming? If I would like to ask you if there is someone with a question, that please raise a hand or just write in the chat so we can organize these. We have a question here already on our side from Priscilla Olsen.
I have three questions. One is actually kind of linked to Lucas' question. I was wondering if you checked on the first set of data, the first part, if you checked for IgG, but in the serum specific to translocating bacteria. I mean, you checked IgA and IgM in the gut, but you know that there's some bacteria that translocate and they're kind of linked to IgG. Some of them are linked to IgG. So there is any increase in IgG specific to these bacteria in the gut?
Well, we would expect, because that's the thing. Like, somehow the impact in the IL-10 deficient mice, it's something more or less well known. Like, it's an old model that people has been studying for a long time. So what was our main focus in the very early part of our project was just to be sure that what is happening to them was really IBD. And by all parameters, we can tell that they are having IBD. But analyzing in more details the inflammatory and immune response in the IL-10, it's become like a second or a third priority. So the priority now would be to understand why MIF is more knockouts are more tolerant. And basically what we discuss, we might be wrong. I mean, you might be right that
eventually the MIF might have even more in the serum. We don't know without doing the experiment. But our expectation is that they would have less, both in the guts and in the serum. They are starting to show a very early bacterial or epithelial damage, and starting the process, one would expect that they would have also more in the serum, but we haven't looked to that.
The other question was also concerned the MIF part. If you have used, and I don't know if you mentioned this, if you used that training bacteria that are not actively doing anything, is it just the antigens that do something and and are transferred, and then the immune cells are doing something because the antigens are there, or is it the bacteria that is actually actively inducing the disease?
Yeah, I'm not sure whether we did that. I will ask Denise. It's super simple. It's just to hit the phase. I'm not sure. I would say that we haven't done that, but we should do that, definitely. But I think that we haven't done that.
The third question was actually about the COVID part, which was have you checked for MIF?
We haven't, but other people has been looking to MIF, and it seems to be increasing in the severe patients. But we should have done that.
You guys have a question on your side? Gustavo, Luísa, anyone else?
Eu tenho uma. Se puder já pular, não estou vendo nenhuma no chat. Sorry. So I don't see anybody raising hands or writing anything on the chat.
Sure, go ahead.
Okay, Marcelo, first of all, super thanks again. Your work is always brilliant, as well as your talks. And because I really like a lot of your philosophy, I think I'm going to ask one more philosophic question, as whether or how do you think these disease tolerance may have shaped the evolution of populations?
Yeah, I mean, that's a point, right? Like, if you see the concept itself of disease tolerance is that it is able to impact the health without affecting the presence of the bugs. And I don't agree with this simple statement, because I think that disease tolerance will impact in the communities and in the way we deal with pathobionts and the way we deal with pathogens. So if we think that we are not just the proteins that are encoded in our genome, I believe that the disease tolerance mechanisms were absolutely and are crucial to live together with our microbiome. So it has a profound impact in life the way we see, from plants to vertebrates and primates and so on. So I think it's absolutely fundamental. The same way that resistance is also absolutely fundamental to life the way we know. And the third one that I didn't mention is avoidance. Let's say using masks is a way to avoid being infected, or smelling the smell of a food that is full of bacteria, fungi, is also a mechanism to avoid infection. So I think that these three mechanisms are absolutely essential and shape the way life it is on Earth in terms of the multicellular animals and also plants.
Right. But I was more thinking about when you did this historical of humanity. Okay, so era pre-antibiotics or so on and so forth.
Right.
There was no masks and there was no drugs. So, and we always thought that the resistance would play a more dominant role in the host pathogen interaction. But your data showing that the disease tolerance by maintaining a, viral or bacterial load and be able to transmit to a person, an individual in the community that doesn't have drugs or any. So it looked like that would force or make a strong pressure for the resistant.
Exactly.
Okay.
Exactly. And that's the case. Let's say when you think about these asymptomatic carriers, and that's very true. And also, again, focus in your question. There is some controversy related to the populations with sickle cell disease being protected against a plasmodium infection. Studies from Miguel Suarez and other groups that suggest that these differences not only related to the ability of plasmodium to infect the erythrocytes, but also the way that sickle cell individuals, by having more hemoxygenase, would deal with infection through disease tolerance. So we have more individuals with sickle cell disease in the areas of a plasmodium, and people think that this is at least in part also due to disease tolerance mechanisms.
Thank you very much. Just one thing to add is, so you show, well, a few situations where the pathogen load is the same, okay? But is it, I imagine, I just want to hear from you that there are other situations that the disease tolerance is also accompanied by a resistance. So you would have different, loads.
Absolutely.
Right.
Yeah, absolutely, Gustavo. That's more or less what I show in that 4 major possibilities when you have, like, in one individual, high tolerance and high a resistance versus like low tolerance and high resistance and so on. So I think that in each individual, these 2 main ways to deal with infection will play a very crucial role also in the communities of infectious agents that we have. As we showed, like, in our case, we are looking diversity. So in those studies, they were looking just the pathogen itself. So in our case, in the toxo model, we show that the pathogen is increasing in the animal that has more a tolerance, but that could be due to lower resistance. So I think that the combination of 2, it's really important to define these and the community and the disease.
Obrigadíssimo. Thank you.
So we're going to take one last question because we really run over our allotted time this time, and it's once again would like to thank Marcelo and for this very exciting talk. That's why it took so long. Very nice data, and we have a question from Professor Julie Shamstein that has to come near the microphone, please.
It's okay here?
Sure.
Can I sit down?
Yeah. You can sit down, please.
So it was great to hear your talk. The second part, I think that we need a forum on COVID that
Yeah.
would be a good idea to see,
Absolutely.
we get everything together one day, because it's impossible to discuss so much. But I understand your rationale to put it together. Regarding the first part, I think that maybe in the direction of Priscilla, the diversity is an interesting aspect that you see there, right? How is the repertoire of even IgM, okay, in these situations? Because, I mean, perhaps you could expect that, the resistant MIF have a very broad repertoire. So they have evolved, maybe even taking advantage of IL-22 because they have a better barrier. They could afford expanding. And if you have expansion, then you could have more diversity and cross-reactivity. So cross-reactivity could be evolutionarily favorable for them. So this is one question. Related to that, have you ever measured the overall translocation of the bacteria in these three contexts? Because that could fit, to the first scenario that we are discussing, right? So these are issues. I mean, it's very important. You have made an experiment that was so frustrating. You tried so many antibiotics and you didn't remove, the transfer of the pathogenicity. So there's something there, not virus, that is contributing to that factor. Could be a factor that is expanding the diversity. I mean, for example, when you treat with antibiotics, the first experiment probably that you have done, I would believe that you probably would reduce the IBD, right? So you can have one potential pathogen that actually is diversifying for some metabolic reason, and they don't have the IL-22, they are not adapted, to deal with that. So you would have a more diversity, and the diversity will, I have advantage.
One thing is that the kinetics is really short. Like they start losing weight with 7 days. It was really a surprise to us because IBD in IL-10 kept in a dirty facility, it takes like months, like 1-2 months. And in this case, when they were together, it was really a very acute response, like with 7 days, 10 days, with 14 days, all that. So it was a really fast inflammatory response.
Marcelo or all male?
No, we had all the combinations, male, female, they both die similar. That's important.
IL-22 doesn't drop, when you make,
In the IL-10 deficient mice. So as I said, at a certain point, it was not our main priority to really understand exactly what was going on with the IL-10, but identify the bug that was transferring the disease and trying to understand, as you mentioned, what MIF has that would maintain this huge diversity and would maintain them with so many pathobionts and not having a disease. So that's our main question right now, is to understand what MIF has. So we don't know the repertoire of B cells, and I think that is an important and interesting. But again, I think my prediction is that they will have some parallel to what we see in the gut systemically. But I might be totally wrong. I mean,
the IL-33 is an interesting tip, right? Because so many parameters will not change, but that won't change.
Exactly.
Right?
IL-22. Sorry to interrupt. Just to confirm what you said, IL-22 we saw was one of the very few cytokines that was hugely increased in the Toxoplasma gondii model. So MIF will have a lot of IL-22 upon our infection of toxin.
And for 33, is a good experiment. Excellent. Prof. Marcelo. So,
Yeah.
I mean, it would be an interesting, easy question, just
Sure.
to make the totally
Yeah.
blue and see whether you have, because in terms of vascular growth factor, all repair,
Yeah.
all that cascade, could be involved. And it's a question that we always have an interesting on this.
Yeah, we have like the L22 a blocking antibody. So that's something we're gonna do.
33, I'm talking.
Yeah, I know. We already have the 22, but the 33, I think is really an important one.
Yeah.
Marcelo, thank you very much.
Thank you.
No, it's very exciting. There are so many more questions, but we need to cut at some point. This point is as good as any. We can continue this discussion here. And people, if you have any more questions to Marcelo, please feel free to write him, to call him, to send smoke signals.
Right.
Marcelo is everywhere. So thank you very much again for the attendance. Today we are going to have a new seminar next week. I don't know if we have already the title for next week.
Luis, yeah, I don't remember now, guys, but we will submit, these few days.
No, sure.
Next
I am sorry.
I should have known. I should have known, but I'm confused now. So maybe tomorrow we already send the advertisement. Okay, guys.
Sure. Thank you very much.